Hi everyone. Uh, I hope everyone is doing fine. Uh, and in today's class, I'm going to talk about immunohistochemistry. Before going to talk about immunohistochemistry, uh, is anyone has any experience working in the lab, a research lab, or anyone has experience doing the immunohistochemistry? Uh, this immunohistochemistry is a very powerful tool, technique used by the research scientists as well as the uh, some of the diagnostic lab people. They use it for the diagnosis of different kind of disease like cancers uh, and other kind of disease. So let us talk about what is immunohistochemistry, the principle behind the immunohistochemistry, the methods, how the methods used in the immunohistochemistry, as well as some of the basic steps. So this is a basic introduction about the immunohistochemistry. In this class we are going to talk about. So first of all, the introduction of the immunohistochemistry. Immunohistochemistry is, as you see, the name itself clears. It is combinations of immunology, histology, and then chemistry. So immunohistochemistry combines with histology, immunology, and biochemistry for the identification of the special tissue, special cellular component in the tissues by using the specific antigen antibody reactions. And the antigen antibody reactions then is visualized to show the distributions and the localizations of this tissue component. So this that what is tissue component means, it could be a protein or part of the protein or whatever. Basically the proteins. So in immunohistochemistry can be used either in a cell grown in a slide, or it can be used in a tissue sections. So the tissue sections can be made by using the paraffin embedding, or by making frozen sections, or also could be used the vibratom sections. So in the tissue sections, we have, beside the cells, we have some of the extracellular components are also present in the tissue sections. So what is the principle behind this immunohistochemistry? So as I already mentioned, it applies histological technique, it applies immunological techniques, or it studies the chemistry also. So the principle of immunohistochemistry is to localize a particular antigens or protein in the tissue sections by the use of label antibodies, and that the interaction between the antigen and antibody are visualized by using a marker such as like the fluorescent dye, or enzymes, or radioactive elements, or even the colloidal coals. So these are used. So let us talk about the methods in immunohistochemistry. There are two methods are used basically. The first one is direct methods, and the second one is indirect methods. So let us talk about the direct methods. So in the direct methods, there is only one antibody is used, which is labeled, and there is a tissue antigen. So the tissue antigen is bound with a label antibody, and this label antibody has already been tagged or labeled. So we just get the signals. It is very easy, very quick techniques, direct methods. But the problem with is, since one antigen bound with one antibody, So the signals getting is very low and very weak. So this is not that much useful in terms of quality, but it can be done in a short time. So some people also use it. And then the next one is the indirect methods. And in indirect methods, there are two antibodies used, two layers of antibodies is used. So this is the tissue antigens here, right here. And the primary antibody, which is on label, which is bound with the tissue antigens, and the secondary antibody, which is labeled with the fluorescent dye or the enzyme or a reporter molecules. So the primary and secondary antibodies, they come from the same species. So the primary and secondary antibodies, they can bound to each other. Since there are the numerous sites in the primary antibodies for the secondary antibody, so one primary antibody can bound with a multiple secondary antibodies. So the signals coming from this indirect method is very good. So this is very often used these days. And now let us talk about the steps in immunohistochemistry. There are basic steps in immunohistochemistry which everyone follows. So the first step is the fixation of the tissues. So the tissue should be first fixed, because the tissue architecture and the cell morphology has to be intact for using the immunohistochemistry. So fixation is done for that purpose, and then formalin or paraformaldehyde solutions are often used for the fixation of the tissue. And then second step, the tissue has to be cut into the thin sections. So in order to do that, we have to make the tissue hard enough to cut into sections. So it is done either by using the paraffin embedding, or it can be done with making the tissue frozen, or it can be even used by using the vibrotomes. So sectioning is another step. And then third step is blocking. So to prevent unnecessary background staining, we use some of the blocking solutions. So the other step is the staining. So antibodies are used for staining, as I already mentioned, primary and secondary antibodies. And these antibodies are wash out. We have to wash out the sections frequently, but to get rid of these unbound antibodies. And then the final stage is mounting and microscopy is done, and view in the microscope. So this is a section. This is a sample in which I have done the immunohistochemistry of the spinal cord sections, stained with GFAP. And the application of the immunohistochemistry, it can be used in the research lab, or also in the diagnostic lab it can be used. So that's it for today. If you have any questions, anything like that, you can ask me or send me an email. Thank you very much.